Nothing earth-shattering, nothing earth-shattering here, but it's just a nice way to kind of give an overview of what we do. Um, let's see, but before I start with the Oyster stuff, I feel like it's always important for me to talk a little bit about marine geo. And this is the program that I work for. And so GEO stands for Global Earth Observatories. And so um, the idea is that we're focused on coastal biodiversity and in general foundation species. And so how they're changing with changes in the environment. And so tracking long-term change. Um, the program has been around for, I think we're in our uh, over 10 years now. We've been collecting data in a variety of different habitats in the lagoon and then all around the world. So as I mentioned, we're really focused on coastal biodiversity. That sets us apart from a lot of different observatories where they're mostly interested in physical uh, characteristics of the ocean. We are focused primarily on the flora and the fauna. So our research themes are pretty broad, but in general, we're curious about how marine ecosystems work. And so when you start to work on a global scale, the first thing you realize is that we really don't know how these systems work. We know how they work locally. I know how my backyard works. I know how my neighbors' backyards work, but we don't know how that compares to different places around the world, even along the same coastline. So North Atlantic versus South Atlantic, and so we're learning a lot of those things, and we're trying to make sense of that all. So we're interested in how these are changing, and so we're going through rapid environmental change, as we all know, and so we want to know how those are changing and how to keep them functioning. And so we think a lot about ecosystem functions, ecosystem processes. Um, the habitats we work in are generally going to be coastal, so we work on emergent vegetation, so mangroves, salt marshes, those types of things. We work in submerged vegetation, so we do a lot of seagrass work around the world. We also work on hard bottoms, and so we study corals, the fishes around those corals, rocky intertidal, those types of things. And then we do spend a lot of time with artificial structures, which is actually my expertise. And so I spend a lot of time thinking about everything that humans put into the water, seawalls, marinas, docks, all of the animals living on those and how they're affecting the ecosystem. So that's what I spend a lot of time thinking about. But thankfully, through this program, I get to work in all sorts of different habitats, including oyster reefs. So we do a couple of different things. So the first thing we do is we do long-term monitoring of the coastal habitats. And so this isn't super exciting, but over time, you can develop a really nice baseline on how these systems are changing. And we do that through a couple of different ways that I'll talk about in a second. The other thing that we do that we're really good at, and I think what makes the program really exciting is that we do these coordinated regional and global experiments. And we do something that's relatively standardized and we do it here, but we do it all over the world. And so we can ask really large questions using relatively simple methods, looking at whether it's just fundamental ecology, so basic sort of theoretical stuff, or we can look at more mechanistic kind of drivers of change. So if we're looking at our monitoring data and we're looking at why things that are changing, we can start to ask, well, why are they changing? How can we manipulate the system on a large scale? And we do those types of experiments. So every year we'll do one or two of these different types of experiments. Um, this map is showing you all of our partner sites. And so the yellow dots are the partners that have participated in our global experiments. And so these are typically one-off experiments. The red dots are going to be our long-term partners. And so these are partners that are doing the monitoring with us, and they're also contributing to some of these experiments. And so we have a pretty large global reach, which is nice. The last thing we do when we're talking about standardized data is that we have to deal with that data in some way. And so we've gotten pretty good at how we store data, how we do our QA/QC, how we synthesize that data to tell our narrative, and then how we take that data and we turn it into actionable science. And so how can we make a difference with all of this data coming in? And that has been a struggle to figure out how to do all of that in a really standardized way. But we've gotten really good at it over the years, and it's one of our superpowers. So in terms of standardized monitoring, one of the things that I spent several years doing is writing these different protocols. And so we want to do everything the same way wherever you are in the world. So if you're looking at oyster reefs here, how do you do that uh, in New England? How do you do that on the Pacific coast where they have a completely different type of species, or in Europe, or in Hong Kong, or in Australia? So we have to write our protocols so that they make sense both locally, but they can also be applied on a global scale because that's what our mandate is. That's what we're really interested in is these big changes. And so we have to figure out how we can do both. And so we have protocols for seagrasses, coral reefs, the fouling communities, which are the groups of animals that live on artificial structures that I focus on. Oyster reefs, we have kelp protocols, we have fish protocols, soft sediment protocols, lots of different protocols for studying basically any habitat that's in the ocean. Um, so the way that we've structured these is that we follow this GOOSE system, which is the Global Ocean Observing System. It's a group run by the UN, and they have these guidelines on how you study the coastal systems or even the ocean in general. And they've come up with these EOVs. And so these EOVs are essential ocean variables, variables that you want to measure if you're doing a long-term monitoring that tell you something about change, important variables. But when you really think about it, these are things that a normal ecologist will go out and sample anyway. So composition, abundance, density, demographics. So if we think about seagrasses, we want to know the length, the width, those types of things, different characteristics. Spatial extent is a big one. So we do that with all of our different habitats. So the area coverage and how that's changing. And then something simple like biomass, which is a really simplistic metric, but it can tell you a lot about the system and how the system is changing. And so we try to incorporate all of these into our monitoring protocols so that we can look at cross-program synergy, so cross-habitat synergy. So if oyster reefs are changing, does that our seagrass is changing too? So we have that kind of data measured in very similar ways, so we can ask those types of questions. So moving to the topic, so oyster reefs in the lagoon, and so we started to do this in 2017, and so I wrote the our protocol based on this book that NOAA came out with the help of TNC and some other groups, basically habitat monitoring for oysters. And I didn't want to reinvent the wheel. There's no reason to do that. And so what we did was we took it and I kind of restructured it a little bit. And we did a lot of field testing. And so that's always important because you can read something and then once you try to go out and do it in the field, it's always different depending on where you are. But this is all based on NOAA guidelines. And we've incorporated our EOVs into that so that it's globally accepted protocols that anybody can use. So the things that we measure, which I'll show you data for, I'm not going to show you data for all of these different things, but the things that we do on an annual basis, so every year we go out to our sites, is we look at reef extent, so the aerial extent of the reefs, the height of the reefs, percent cover of what's there, so we get an idea of what the reef looks like. The density of live oysters, the size frequency. So that's going to be pretty important, and I'll show you why in a second. Rugosity, which I'll get into, and then the thing that I think I'm most interested in, and I think is really important that most people don't look at, is the associated biodiversity. So all of these foundation habitats are providing home and refuge for a lot of different species. And so when we lose these habitats, one of the things that we need to understand is that we're losing a lot of these other species too. And so there's a lot of species that like to live in these oyster reefs. And so we want to measure the abundance of them, the richness of them, the diversity of them, and how those species are changing over time and in different types of reefs. So pristine reefs versus not so pristine reefs or restored reefs versus natural reefs. And so those are the types of questions that we really like to get into because they're really interesting and they can tell us a lot about how the system is functioning. So these are our three sites for those not familiar. I think everybody's pretty much familiar with the lagoon. We're right by the Fort Pierce Inlet. on the eastern side of Florida. So that red star, that's where the Smithsonian Marine Station is in Fort Pierce. These are our three long-term sites. So there's Jack Island, Wildcat 1, and Wildcat 2. We initially started with, believe it or not, I think 8 different reefs in 2017. Um, and as the years progressed, these reefs got worse and worse. And they got to the point where we couldn't even go out there and lay a transect and find oysters anymore. So we've stopped going out to those reefs because they're effectively extinct at this point. So these are our last three relatively good reefs, but as I'll show you with some data, the reefs are not doing that well either. Um, so when we go to a reef, the first thing we do is we lay out transects. And so this is relatively common. And so depending on what your reef looks like, we can have 3 transects on a single reef. If you have patch reefs, you can add different transects to them, but we all try to keep them about 30 meters long. And so when we lay out our transect, we really want to have a decent looking reef. We don't want these small, tiny reefs, even though sometimes that's all you can really find. But the bigger the reef, the better. And depending on the size, we can manipulate our transect tape to fit those reefs. And so this is the first thing that we do when we go out to a reef. So getting into some of the data, and I'll go through kind of the methods a little bit, but I think I'll do this as I'm going through some of the data. So this is percent covered data over since 2017 at our 3 sites. And so the way that we do this is we lay our transect tape out, and then every couple of meters we'll put down a one by 1 meter quadrat, and it's got 81 intersecting points. And you can see the picture of it there. And under each point, we just say what's there. And so whether that's sediment, whether that's live oysters, whether that's culch, which is going to be just a half shell, whether that's box oysters or whatever else is in the reef, whether it's rock or algae or whatever we find. So this is a really nice way to just characterize the reef. And so we have 3 transects. Along each transect, we put down 5 of these quadrats. And so we have 15 different measurements for each reef per year. Um, so as you can see, which is relatively disappointing, is that the culch is increasing over time. So this is just the shell on these reefs. And that's in pink. And the green is our live counts. That at all of our sites is going down. Our all of our sites are declining. And so one of the things that I'm really interested in working with this group is that I'd like to know some baseline numbers. And so maybe not today, but to continue the conversation of what we do as it compares to other sites in the lagoon and other sites around Florida, what's the percent cover of everybody's reefs? How does that compare to our reefs? Because I don't know. I don't have that information, and we haven't been working with this data long enough to kind of explore those different trends. And so I'm not sure how this data compares to everyone else's, but the one thing that's pretty visible is that our oysters are not doing so well locally in the lagoon. And there is some variability in that, but in general, we're seeing declining trends at all of our sites. So one of the things that we measure is size frequency. And so along our transects, what we'll do is we'll lay a quarter meter by quarter meter quadrat down, and we'll excavate everything in that quadrat. And we can get a lot of interesting data from that. We can get the size of live oysters, which is really important. We can also get the density of live oysters, which I'll show you in a second. So in terms of size, we measure from umbo to tip in millimeters. And this is just showing you the size distribution of oysters over the different years. So the different years those are the different colored lines. I don't have a legend here because it doesn't really make sense to look at individual lines. But what we're seeing is that our oysters are small. They're at Jack Island, they average around 40 millimeters. At the 2 Wildcat sites, they average around 45 to 50 millimeters. And this is really important. And one of the things that I'm really curious about is why this is. Why are they staying so small? The harvestable size of an oyster in Florida is 76 millimeters. It is almost impossible to go out to these reefs and find an oyster that is 76 millimeters long. And so I'm very much curious on why they're staying small. We have some ideas why that is. Some of us, after chatting, having long conversations about this, is that, well, the waters are warmer. So what does that mean? That means that the oysters are putting a lot of their energy into reproduction and not into growth. So these oysters are staying small, but they're reproducing all year round instead of those two seasonal peaks that we're used to. I think in Florida, we're used to seeing them in early summer and maybe in fall when those temperatures hit the perfect mark. Instead, from some other studies that we do, we're seeing recruitment kind of year round. And so I'm curious whether this is just a change in the temperatures of the water. They're staying small, more energy is going into reproduction and not into growth. Um, but that remains to be seen, and I'm not quite sure how we can effectively test that and get a definitive answer. But I think the take home is that our oysters are small. And they're staying small. And at some of the sites, they're smaller than others, which I think is a really interesting trend, um, and something that we definitely need to do a little bit further work with. So as I had mentioned, we also count the density of oysters. And so within that quarter meter by quarter meter quadrat, we count all of the live oysters that are bigger than, I think they're bigger than 10 or 15 millimeters. Once they get past that, then we know that the survival is going to be much better. If they're smaller than that, we typically don't count them because the chances of the survival is relatively slim, and we don't want that to inflate our numbers. Um, but what we're seeing, at least at two of our sites, at Jack Island and at Wildcat 2, we see declining trends in oyster density. We do see an increasing trend in the middle graph at Wildcat 1, um, but it's just we're still just seeing declines in overall oyster percent cover over time. And so again, this data is not it's it doesn't make me happy to see this kind of stuff because we're taking multiple metrics of these reefs and we're just seeing declines everywhere. Now there is some variability in this, but overall the trends are significant and the trends are declining besides this one site. Um, but again, this tells you a lot about how your reefs are behaving. And through these different metrics, we're just seeing declines. So one of the things that we're really interested in is we're interested in rugosity. And rugosity is basically a measure of complexity. And within the marine environment, within any system, we know that complexity is really, really important for a variety of reasons. Think about a coral reef. That's probably the best example. Coral reefs are very complex habitats, and that complexity drives diversity. There's been hundreds, if not thousands, of studies that have looked at different habitats, their complexity, and how that relates, how you model that with biodiversity. And we know that the more complex a habitat is, the more biodiversity it will hold. Well, what's nice about rugosity is that it's really easy to measure. So we use a chain and tape method, and it tells us a lot of different things. It tells us first, are our reefs becoming more flat? And if they're becoming more flat, that means that there's not a lot of live oysters left. Imagine a healthy reef versus something like a sand flat. Like that's kind of what we're getting at here. Um, the other thing that we can do is we can use rugosity to predict or we can model associated biodiversity. And so the more rugose or more complex a habitat is, the more associated crabs and shrimp, amphipods, polychaetes, those types of things you're going to find in your reef. I'm not going to present any of the modeling data, but some of our data suggests that this is a pretty good metric to look at the health of a reef. And I do have colleagues in the Gulf who will just go out and measure rugosity. And that's the only kind of data that they're going to take because that can predict the health of the reef. So they just go out with this chain and tape method and they take 15 or 20 measurements of a reef, and that tells them a lot about how the reef is behaving. And so the way that we do this is we just have a meter long chain and we overlay it over the oysters. And there's a figure showing how this is done. Um, And the plots, so this is the inverse of rugosity. So we take a 1 meter chain, and when we overlay it, it becomes smaller. And so what I've done here is this is just one minus that number, because the way that at least I think is the bigger the number, the better the metric. And so as this rugosity measurement gets higher, that means the complexity of the reef is larger. And so that's one way to think about this. Now, what we're seeing here is that in all three cases, like all of the previous graphs, we're seeing rugosity get smaller. So the reefs are essentially getting flatter. And so that will tell you that, well, there's not a lot of live oysters left. It's becoming flatter. It's just really layers of shell, of culch shell. And so one of the things that we can do from a statistical point of view, from a quantitative point of view, is we can start to model this with different characteristics of the reef, and we can show quite nicely that rugosity is a really nice predictor of the health of the reef. And so it's something simple that you can go out and do and measure, and it can be really, really informative. And so we like these types of data. We like to go out and collect something simple that tells you a lot about what's going on. Um, One of the things that we want to do that we haven't really done yet is do kind of drone work on these reefs. And with a drone, you can easily fly it over and you can get complexity, rugosity measurements, but you can also get spatial extent. You can a lot of different things with drones. We haven't got there yet, but that's next on the list of the things that we want to do. One of the things we can also do is we can measure all those little fringe reefs that we have around the area, to look at some other different sites where we might want to be interested in how these reefs are changing. Um, but for now, this just chain and tape method is really, really useful. It's well published in the literature so that we know it works, and it's you can get a lot from it. And so we like to use it. So the thing that I'm really interested in is biodiversity, and so how these habitats are providing home to different species. One of the things that we've developed are what I call bio boxes. And they're basically, it's a box that you put out into the reef, and you excavate a small portion of the reef, and then you fill it with shell, and you leave it out there for 2 months, and you go back and you take that box and you put it into a tray, and you just pull out all of the associated fauna. And so it's a really nice way, very easy way to look at how these reefs are providing refuge and habitat for different species. And so here, just our three main players that everybody should be familiar with if they work with these species in the reefs, the Eurypanopeus, Panopeus, and then the Petrolithes porcelain crab. So these are just some plots of the different species over time. Um, Not a lot of trends going on here, so the data is kind of messy, but we can see when there's good years versus when there's bad years, and we're always curious. For instance, in 2023, why did Petrolithes do so well? And so these are data that we can go back and look at. I think we're going to need a lot more years of data to start to look at these, but it just shows you, I mean, overall, it looks like there's some declining trends in there. And so that makes sense when you think about comparing pristine reefs versus reefs that are starting to be degraded. And so that's an interesting scientific question that we can pull out of some of this modeling data. We can model that and get some interesting answers. And so how do these reefs perform if they start to become degraded over time? Are they still providing the same sort of resources? Presumably not. These crabs are, I wouldn't say they're obligate with live oysters, but they like to be where the live oysters are. And so if we're just finding shell, these crabs, the numbers of them should sort of peter out. And so I don't think anything significant here, anything earth-shattering, but again, it's a really nice way to look at how these crabs are functioning um, within the oyster reef community. So, as I had mentioned, we are really interested in in scaling up. And so, how do the lagoon reefs compare to different reefs around the world? When we're thinking about Crossostria, of course, we're thinking about the East Coast and the Gulf. And so, um, one of the things that we did in last summer is that we took our protocol, just our standardized methods, and we found a bunch of partner sites who wanted to do this as well. And we did it at all of those sites. There were some minor changes to make it a little bit more easier for some of the partners to do. But in general, we did all of the same types of things. The red dots are going to be the intertidal sites. The blue dots are going to be the subtidal sites. But the things that we had 19 different sites and we had 54 different reefs that we measured, which is pretty significant. There's I can't think of another study that has looked at just basic characteristics of oyster reefs along a latitudinal gradient. And so that's the first thing that we wanted to do before we started to pursue other more interesting ecological questions, is we just wanted to look at how these reefs are, what are the characteristics of these reefs over scale. And so we measured the different environmental factors, and so temperature, salinity, turbidity, all those types of things. We measured the reef height, the density of the oysters, the shell length, diversity, and we tried to do some recruitment work as well. So we could kind of get a really good idea. It's very broad, brushstroke, 20,000 foot view. These aren't detailed surveys, but it just gives us an idea on how these reefs are functioning. And so these are the types of things that we're really interested in. How do our reefs compare to other reefs locally, regionally, or across latitude? So I'll show you a little bit of the data that's been worked up so far. There's still a lot to go through, but this could be interesting to some people because it's intertidal versus subtidal reefs. We don't know a lot of those differences, and so this starts to get at that. But also we're looking at latitude, and so the first graph is just oyster density. So how many live oysters are you getting? And this is scaled up per meter, and so we're seeing differences within intertidal and subtidal reefs. So in the intertidal, the densities are much higher. And remember, this is done across latitude. So this isn't just the IRL or the Gulf. This is modeled at all of our sites. And so this was significant. And so that's an interesting result that really hasn't been looked at before. And so why that is, these are types of questions we can start to focus on. Another interesting question that was easy to model is just the shell height. And so how long are these shells and how does that compare in the intertidal versus the subtidal and across latitude? So on that x-axis, it's 0 to 40. So as you move, so 0, of course, is the equator. And as you move up, you're going to the northern latitudes. And so we can model that, and we can model that how that compares across subtidal versus intertidal sites. And so the couple of cool things that we found so far is that the oyster length, I'm going to use length even though it's written here as height, stays relatively stable in the subtidal zone, and it's quite large. And so even the first graph is showing you that the density may be smaller, but the oysters in the subtidal area are actually larger. So that's a really interesting result. In the intertidal, they're smaller in length, but as you go from warmer waters to colder waters, those oysters get bigger. And so that's another interesting result that we did not expect, but it's something that we can pull out with some of this basic monitoring data. And so it looks like as you're getting close to 40, 45 degrees north, the oysters are becoming the same size between the subtidal and the intertidal. And so that's a really interesting result. So why are they smaller in warmer waters where you would expect them to be larger? Because there's more, the waters are warmer, they can metabolize a lot faster. There's presumably a lot of food down here, so productivity is higher, but it's an interesting result that we did not expect. And then some basic results with the associated species. And so initially we looked at faunal richness. So richness is just the number of different species you have in your bio box. And so that decreases as you move from north to south or south to north, which makes sense because there's a latitudinal gradient in biodiversity. So as you move from the poles to the tropics, there's more species, and this is a global result that we've seen in all different types of habitats. And so we expected to see that, but it's nice to see that and oyster reefs too. However, the one caveat is that we're dealing with only a few species. And so uh, six average species, and we're going down to like three or four in the northern regions. And so not a huge difference, but still the trend is showing us that as you move to the north, you're getting less species within these oyster reefs. In terms of intertidal versus subtidal, not a big difference. And so we're seeing the same amount of species richness in the subtidal versus the intertidal. And then one of the things that we started to kind of think about is differences in the East Coast versus the Gulf. And so they're effectively they're separated from each other. And so we're starting to look at how those reefs are different. In terms of associated species, well, we're not seeing a lot of differences um, in the species richness, but we have abundance data. We have a lot of other data that we're going to start to go through. But I think some of these initial results are quite exciting because this really hasn't been done on scale before, and so it's going to give us a lot of ideas to move forward with, so we can start to figure out what's going on in these reefs. How are they changing over time, not just locally, but on a really large scale, just using really simple methods. I mean, it doesn't take very long for us to go out to a reef and get all the data that we need. We can do it in an hour and a half or 2 hours. And so it's one of the things that we like to try to get our partner sites involved with. And so one of the things that we do is we have an Oyster working group where we meet monthly, where one of the focus is trying to get other sites involved and to do our protocol. And then all that data goes into the Marine Geo centralized database, and anybody can use it to ask different types of questions. And so we have a lot of postdocs and PhD students who like to take that data and turn it into a narrative and start to look at it and to see what's going on. Um, so right now we're spending a lot of time figuring out how we can get these sites that participated in this network experiment, how we can get them to become long-term partners. What are the carrots that we can provide to keep them coming back? And whether that's us publishing papers, which is something that we're really good at and we spend a lot of time doing, or it's sharing data, those types of things. And so it's part of what makes a network work is getting everybody to collect standardized data so that we can look at these really big changes over time. And then do a couple of these types of experiments where we can look at just one detail of what's going on, whether that's the length of an oyster, whether that's the crabs living in there. One thing that I forgot to mention that I do with all of my data is that I look at all the parasites in the crabs. And so when we're collecting these bio boxes, we get lots of crabs in there. The petrolithes and the panopids, they have the barnacle parasites, isopod parasites, and so we count all those and we sex all the crabs. And so we have data on that too. So we can look at how parasite loading is changing in the different types of reefs, whether it's pristine, restored, natural, degraded, and how that changes across latitude. And so just collecting a lot of really simple data on these reefs can tell you a lot about what's going on, but it's got to be done over time and at a lot of different scale on a large scale. So that's all I have for this. It was Vincent had asked me to do this, and I was like, well, this is a great chance for me to start looking at some of this data. And we were waiting sort of for the 10-year mark to start to write all this up and get it out there. But this has given me a lot of thought for moving forward with writing a lot of this stuff up and trying to make sense of it, model all of that. And then lastly, all of this work could not be done with the help of our colleagues. And so in particular, people at the DEP have been, with the Aquatic Preserves DEP, have been working with us since day one. And they're always willing to come out and help and bringing interns and bringing volunteers and whoever. And so none of this work could have been done without their help. And so a huge thank you to Matthew Anderson and Emily Sermont and everybody at DEP who uh, will help us collect a lot of that data. And so I think that is all that I have.
Thank you, Dean. That was
Sure.
that was that was excellent. Um, there are a few questions in the chat, and I'll go through them.
Yep,
And then please, any if anybody else has more questions, at you can either enter in the chat. I also feel free to unmute yourself and ask directly.
sure,
Um,
please. Yeah,
I got a couple of questions, but I'm gonna go pick the other ones first in the interest of time. Nikki was asking, what time of year do you monitor, and when do the oysters spawn in the IRL? I guess particularly where you're sampling them. Lorraine answered that the spawning is typically April to September. Um, but what time of year are you typically doing this protocol?
so we're dependent on low tides, and so the best low tides are going to be late May and June. So we'll find the lowest tides of the year, and that usually is in June, and then we'll do all of our oyster stuff there so that we have the most amount of time on the reef to do our sampling and to do whatever else that we need to do. Um, and it's once a year, which we feel like these oysters aren't changing very much, and so once a year is an accurate measurement. In terms of spawning, yeah, Larray's like right on point with that, and so we typically see them in the summer. I put out a lot of fouling panels to do a lot of other work, and I do that throughout the year, and I'm starting to see them just all times of the year. We can find oysters within below 10 millimeters that we know are a few weeks old on these, and so we feel that the recruitment window is opening up with changes in water temperature, and so we've been trying for years to figure out a very simple way to measure recruitment with oysters, and it's always been some things are a little too time consuming, or you got to go out on the boat and get to these reefs, and it's a lot of effort, and we just haven't found a really good method to ask those types of questions. But right now, I would say I see the most recruitment in June and July, which is unusual because that's when waters are really ramping up, and so I'm used to seeing it earlier in April and then another peak in late August, September, not August, but probably September, October when the water temperatures are going back down. So they're like those two peaks. But now it seems like it's all year round.
Hmm, interesting. There's a question from Lorray. Who is your partner in North Florida for the Latitudinal project? And looks like Nikki answered that. Um, GTM, of
Yeah.
course. And let me see. There's one more. A question from Natalie from Orca. How many bio boxes do you typically put out along your study sites?
So we do 3 bio boxes per oyster reef. And so three is in terms of quantitative analysis, you need at least 3 replicates. Um, so we'll put out 3. We used to have them half meter by half meter, but we've gotten much smaller now to make it more doable for our partners because it can be a lot of work when you have a lot of crabs in there. Um, but we try to we put out at least 3 per site. And we've been adding things like temperature loggers onto those bio boxes and spat sticks and all sorts of different things where we can collect some extra data. We'll put them out in usually early April they'll go out, and then we'll pull them out in June when we do our full sampling.
Cool. So it's a once a year
Yeah,
then? Okay, once a
yeah,
year. Okay,
yeah.
great. Um, just another thing real quick. I put the Marine Geo website address. Is that where we could go to get to those protocols?
Yeah, they have those protocols in there, but I'm also happy to share. We've been changing our website quite frequently.
Oh,
Um,
okay.
but I can certainly send it to you and you can pass it around. It shouldn't be hard to find. If you
Okay.
just Google Marine Geo Oyster or whatever the protocol you're interested in.
Okay. Great, great. I had some problems with our internet here, so I just went ahead and just put the general
Okay, yeah,
website
it should
address.
be in there. Yeah.
Okay, great. Let me see if there's any more questions. And oh, thank you, Nikki. Nikki posted a paper that was done by um, Dave Kimbrough
Hmm.
and Grabowski.
Yep.
Uh, and was that did that all feed into the Marine Geo network? Was that all Marine Geo protocols?
No, no.
Okay.
Yeah,
Oh,
this
this
is
is
something.
earlier. That's right.
Yeah,
Yeah.
this is earlier. Yep.
Yeah. Right. And I'm curious if maybe some of what you're finding, how does that compare with or will potentially compare to
Yeah, we need
previous?
to start to dig into some of that. So we have a few key players in the oyster world in our working groups now. So Jeb Byers and some of those people who really know the systems along the East Coast really well. So we're trying um, to get better expertise uh, in these and synthesize all of this data and come up with different sampling strategies and those types of things to look at some of these questions. But we're trying to, so their paper was a sort of a one-off. We're trying to take that and continue it and do it every year so that we can, because we have the capacity to do that, to continue those types of efforts.
Great. Do you have any more information about the working group? How do we, is this to sort of adopt these protocols to be part of that Marine Geo network or?
Um, So, well, I mean, the working group is typically so we're meeting monthly,
Mhm.
and then it's for Marine Geo partners, so people who are
Mm,
involved in
okay.
doing our sampling, and we get together. And one of the things we've been spending a lot of time talking about is this network project, and so going over results. I spent a lot of time training people on how to do that, so we spent a lot of these monthly meetings on how you go out to a site, how you measure a height, how you tell apart. So I had to put together like a large PowerPoint on the different species. So I do a lot of taxonomy. So who those species are, depending on where you are along the latitude. So it's been a lot of training, and now we're starting to go through some of the data. We have a postdoc who's leading this project, so she's been working furiously with trying to get some of this out and making sure that we get some of this information to our partners so that they know that we're continuing to work on it, write it up, and those types of things. And now we're trying to figure out, um, we spend a lot of our time in meetings now trying to figure out how to get more partners involved with the program and collecting data. So one of the things that we do that I spend a lot of time doing is training. So I go to these different sites, help people get set up. We give them the supplies, and then we go out and we train them on how to do this. And then they become partners, and then they can be, part of these network experiments where we're doing these types of things, and we do all the data handling, all of the analyses, all the writing, all of that. And so we basically just ask partners to just go collect a little bit of data for us using our protocols, and then um, they're part of the family essentially.
Great. So so people can get involved as a partner to
Absolutely. Yeah,
assist?
absolutely. Yeah, yeah.
It
So interesting. One of the things that I did over the summer is we have this program. We call it the 3M program, but we're rebranding. Um, but what we did was I flew in I think 12 different professors from different universities around the Americas, so North, Central, and South America, and I train them on how to do our protocols with the idea that they take this training and they bring it into their classrooms. And so it's basically like a reverse REU with people who've done an REU before. That's a research experience for undergraduates. What we're doing is that I'm training professors on how to do these protocols. They do it in their lab classrooms, and so they do it in a university setting where they'll take their lab class, they'll go out and they'll do their protocols. And so we have two different groups doing related to the oyster stuff. We have two new groups, one in Charleston, South Carolina, and another one in Virginia, that they're doing these protocols now in their labs with their classes. And so that's one way that we're building capacity within the program. So we're getting new partners, but we're also increasing ocean literacy. So we're teaching the professors, and the professors are teaching their students on how to do that. So we've provided all of the classroom training materials, um, and then all of the supplies, and then they go out and I give seminars to their students, and then we train them on how to do that. So they're now effectively collecting marine geodata, but they're doing it through a coastlines to classrooms sort of model, which has been incredibly helpful. And it's a really nice way to get involved with student learning too. So that's another way that we've sort of branched out rather than having individual people go out and collect data. We can get the students to do it now, which is always probably the preferred method because it's creating educational opportunities and we're finding people who are really interested in this stuff and they can take some of this data and just run with it and do what they want with it. And so we're always happy to help with that.
Oh, that's fantastic. So maybe that could we reach out to you as a first step
Yeah.
potentially? Is
Yep.
that the
Yep.
okay?
Yep.
Let me see. Do you mind putting your email in the chat real quick? Are there any more questions from the audience? Thank you. Thank you.
I have a quick question.
Yes.
Dean, thank you
Mhm.
so much. That was
Sure.
awesome. I'm really excited to see where you head with all that data. I know that's going to be a lot.
Yes. Yeah.
The one thing that I was thinking when you were talking about the size of the oyster and how they don't get much bigger, and well, they never even really hit that 3 inches, and the temperature. I'm wondering if there's any way to look at what their food source is in the water column, because I think we've seen that the diatoms and the plankton species have changed, and maybe
Mhm.
that's decreasing the filtration rates of them, which is leading to a little bit of stress. Which, as you move up the coast, depending on the difference in the hydrology, and I don't know, just something that I was in the back of my
Yeah,
mind.
yeah. So we don't really know. So that is definitely I mean, you know the area pretty well. And it's a pretty productive area. There's lots of food. Some of the our better oyster populations are actually in these really muddy,
Mm-hmm.
sediment heavy kind of areas, which we didn't really expect.
Yeah.
Oysters generally like nice flowing water. Um, but we don't know what's so one of the things that I'm really interested in, particularly with this group, are what are your average sizes of oysters? Are they around that same size?
Mm-hmm.
I don't know, and I haven't spent enough time reaching out to people and looking into the literature. But all of our data goes to CCARs. So the state's getting all of our data on this. And I think, I'm hoping that they'll kind of synthesize a lot of the Florida data. Um, and then we'll get some of that information. Um, but I just find it to be really interesting that, it's the harvestable size, we're getting half of that at three of our sites. And um,
Stressed for sure.
yeah, I mean, they're definitely stressed for sure. And just, okay, okay,
I
so,
just pulled
so
up our master
yeah.
spreadsheet and just looked at the average real quick.
So we're
So,
pretty close in terms of both percent cover and size. And so I think that's something, I mean, maybe I don't know if the state wants to hear it or not, but that's something that I think is really interesting. And why that is, why are they staying small? Um, I think from some of our work in Maryland, we have good partners in the Chesapeake Bay, and they can grow 40 millimeters in a summer. And when I told them about my size, they were like, that's crazy. That shouldn't be the case. They see them get up to 60, 70 millimeters within a year, and ours are getting stuck at this 40 millimeter mark. And I don't know if it could be predation too. It could be just the larger ones are getting eaten, but we're measuring the box oysters too. Um, and so we're not seeing a lot of gaping oysters. They're all kind of sticking around that same size. And so um, I think it's a larger sort of ecosystem or sort of climate sort of issue going on, but something that we want to really kind of dig into because uh, it's a really interesting pattern that needs to be looked at.
Cool.
And
I'm
one
getting
one other question. Oh,
oh, go
go
ahead.
ahead.
Go ahead, Lorraine. No, no,
If
go
it's
ahead.
on that, I'm moving like thought processes. So if there's something along there.
Um, okay. Well, Jenny Hansen had asked about the
Yeah.
potential for disease or parasite loads.
So
Uh,
one of the things that I didn't mention is that in our network project, we did collect, I think, 20 oysters per all of our 54 sites, and we're looking at condition index, and we're also looking at parasite loading. I haven't seen that data yet, but from what I know, it shouldn't be a disease issue that's causing them to stay smaller. Our oysters are kind of in a higher salinity area, so if the higher salinities prevent, from what I remember, the higher salinities are keeping that disease relatively at bay. Um, so I don't, it could be that. It could be a lot of different things. We really don't know. And so it's probably, as we all know, we've all heard the seagrass story in the lagoon many times, and so it's multiple stressors at this point that's causing these things to occur. And trying to isolate any single thing is going to be just really difficult to do. So we're something we want to do, which it's there. I'm sure we can come up with some clever ways to start to look at that.
Cool. Nikki brought up the potential for coastal acidification maybe being a factor as well.
I mean, that would cause the shells to kind of break down. I don't know if I'll have to look at the pH. We have some really long-term data of changes in pH, but I just don't know. Yeah, I don't
Yeah.
even I've spoken with some people and we're starting to look at all the sponges, and maybe the sponges are just growing on these older ones and they're causing them to kind of break up and just get pushed around. Another idea that it could be boat wake. And so we have a lot of boat wake around our areas. And so that I think it only takes a few centimeters of a boat wake to really cause damage to a reef. So maybe that's causing these reefs just to not persist over time and keeping those oysters small. So again, it's
Mhm.
like all of these different stressors are really having an effect. And I'd have to think that within the next 10 years, I mean, our oyster reefs here aren't going to be around. So we're starting to think of different restoration techniques that are question driven, scientifically proven. We're not going to throw bags of shell out there because that never works. But we want to look at how we can do this and measure success over time. And so we have a couple of proposals we're working on right now where we can start to do some of more of that work. But effectively, one of our sites, Jack Island, which is like a classic, beautiful oyster reef, it's pretty much wiped out within the past couple of years, and we don't know why that is.
Mm.
It happened right around COVID, where we weren't allowed to go out and sample, and it was really nice one year, and then it was completely shelled the next year, and so we're seeing. Just a lot of our sites go down. And I think this is really important. And it's something I wanted to present at the IRL symposium is that we're spending a lot of time talking about seagrasses and why they're not doing so well. We're seeing all of these other habitats that aren't doing well either. And I think we really need to like bring that into the conversation because it's just an ecosystem wide degradation that's happening, at least in the lagoon. And so knowing a little bit more about, well, oyster reefs aren't doing well either, I think is really something that people need to hear. And we're starting to see that after, X amount of years of data collection.
Cool. Jenny brought up the potential of genetic issues being maybe
Yeah.
smaller
So,
sizes
well,
or begetting smaller sizes.
yeah.
There's selection for that
So
maybe.
that's one of the issues with seagrasses. And so people familiar with seagrasses, we used to have a lot of thalassia in the lagoon. Thalassia, there's only one genetic rennet or whatever. There's only one genotype in the lagoon. We're at its northern range, and so uh, having a lot of genetic diversity is really, really good for foundation species because during those stressful times, you have the right the right genetic makeup to get through those stressful times. And when you have really low genetic diversity, like in thalassia, your system starts to get stressed and you lose it all. So I can go out and find just a couple of leaves of thalassia, but 10 years ago, I could go out and swim in a bed of it. And now it's pretty much gone. So maybe
It's
that's
coming
true.
back, Dean.
It's well, okay. Well, we'll see. Yeah. I hope so because yeah, Halidouli is wimpy compared to Thalassia, but we haven't done the genetics on oysters yet. And so that's something that we want to do, but doing that sort of work is very challenging for a variety of reasons. It's not expensive, it's just doing the bioinformatics is complicated, and you need somebody who's really really savvy in that type of system to be able to do it. in an efficient, quick sort of way. But that's next on our list. That's something that we want to do. We want to do that with the grasses as well. So we have a laundry list of things that we really want to get into. Genetics is definitely one of them, and we think we've thought about that in the past. Maybe it's just these smaller sizes are because of the genetics, but we just don't know.
Cool. I think we got time for one more question that Emily posted here, and she was asking about the presence of Austria equestris. It's a smaller size oyster. Is there potential that this, maybe there's some cryptic measurement of that species that could be affecting those
Yeah,
size?
we don't see, so I only see Austria along like seawalls. I don't see it in our reefs itself. We do see a lot of isonomen species in our reefs, but there's not a lot of competitive interactions that are happening. For the Austria species, when I see it, it's typically further south, so in the Fort Lauderdale, Miami area, and it's kind of all along the seawalls down there. We don't see it very often up here, but it is a smaller species. It's also not a reforming species like Chrysostria. But I don't know if there's competitive impacts that are occurring, but sort of along those same lines, one of the things that we really want to do that we've been talking about for years is to look at all the oysters on these mangroves, because people who work in the IRL, there's a lot of oysters on these mangroves. So we're not recruitment limited, which is the question people always ask. Well, you must not have a lot of recruitment. Well, we have a ton of recruitment. Oysters are everywhere. And so I'm not worried about their function. It's just these reefs are starting to decline. And so how do these reefs compare to like with oysters on mangroves or oysters on seagrasses, and looking at the different measurements? Maybe they're bigger on mangroves, maybe they're bigger on the seawalls. And so that's something that we want to do to look at how the substrate is affecting the size of these oysters too. So there's a lot of questions to ask, a lot of really interesting things that we want to do moving forward now that we have this long-term data in place. So stay tuned, I guess, is probably my best response.
Cool. That's a great way to end it, Dean.
Uh-huh.
And thanks everybody for tuning in. And we'll get back to you when we get our next FORS meeting scheduled. So thanks everybody.
Thank you.